Chenghua Gu studies how new blood vessel networks form, a process known as angiogenesis. Angiogenesis is important during development and wound healing because new tissues need oxygen and nutrients, which are supplied by blood vessels. Here, a network of blood vessels is forming in a developing eye. Angiogenesis also plays an important role in cancer. Tumors, such as the one at the top of the screen, require nourishment, and they can only flourish if new blood vessels sprout to feed them. How does this sprouting process work? Scientists have known for more than a decade that a protein called VEGF stimulates the growth of new blood vessels.
Now, Gu's team has identified how two additional proteins contribute to angiogenesis, offering new therapeutic targets.
Postdoctoral researchers Ji-Ha Kim and Wanjong Oh trace the growth of new blood vessels in the eyes of developing mice, specifically focusing on their retinas. Here is typical blood vessel growth in the retina of a mouse the day it's born. Here's typical growth by day 3, day 5, day 7, and by day 15, angiogenesis is complete. Here's a close-up of one quadrant of the retina halfway through the process.
But here's what happens when the researchers interfere with a protein called semaphorin 3E. The growth is uneven. Some vessels race ahead, while others languish behind. The researchers produced the same unusual pattern of growth when they interfered with a protein that semaphorin 3E binds to, called Plex and D1.
The team showed that together, the two proteins control the architecture of new blood vessel networks by interacting with the famous VEGF, the protein with an established role in angiogenesis. The three players regulate each other and collaborate to tell vessels when they should grow and when they should stop growing or change course. A noteworthy finding is that Plex and D1, shown here in blue, is only produced at the tips of sprouting vessels, unlike VEGF, which is far more pervasive. This localized production of Plex and D1 makes the protein an ideal therapeutic target. It might be possible to disrupt Plex and D1 without interfering with mature blood vessels elsewhere in the body. The team hopes it will be possible to improve upon existing therapies that block angiogenesis. Currently, the drug Avastin is used in many cancer patients to prevent the formation of new blood vessel networks that nourish tumors. Avastin focuses exclusively on VEGF. Future treatments might target semaphorin 3E or Plexin D1 as well as VEGF, increasing efficacy. The study also has implications for treating macular degeneration and diabetic retinopathy, which are both driven by a proliferation of new blood vessels.